So you got them? Oh, yeah, no problem. I'm so sorry. I like couldn't figure it out. No, it's okay. I sent them before.
Okay, just a reminder for those of you who are in the lab. Summary is due, number 2 is due tomorrow. There was a mix-up on the syllabus, but it's 2 weeks after the lab was finished. Okay. Be prepared to answer that question. And this one. Yeah, I will. Okay. So, cancer is a rare condition. Yes, no, maybe, and why? What we were talking about last time primarily was about the checkpoints in the cell cycle and the fact that these can be influenced by outside conditions and endogenous factors like growth inhibitors. RB is that first checkpoint deciding whether or not the conditions are right for the cell to undergo division. That is looking for optimal conditions, but growth factors may in fact stimulate cell division under suboptimal conditions. P53 is involved in DNA damage assessment that can occur under the normal course of events in DNA replication. Why? Why can it occur under normal the conditions of normal DNA replication? Anybody? Why can damage occur to DNA during the normal course of DNA replication? Cell biology, folks. Why is that true? Okay, DNA synthesis is perfect in all cases?
No, it happens only.
Why isn't it perfect in all cases?
You get mutations.
How do you get the mutations? Pardon? Wrong nucleotides? Rarely.
Deletion.
Okay, the lagging strand is a discontinuous replication process. It involves ligating pieces together, and there can be mistakes made, and often are, in putting those pieces together. Most of the DNA damage that actually is being monitored, and why this exists in the first place, has to do with an imperfect synthetic pathway for replicating DNA. Okay. You get, during carcinogenesis, or the development of cancers, you may get failures in those points. You may also get failures elsewhere. Okay. So this is just more on RB. I'm going to skip over that. Okay, disruption of the cell cycle because of damaged DNA, increased susceptibility. Once you get damaged DNA and a cell gets into, or is divided into daughter cells, if there is damage to begin with, the daughter cells are going to share that damage, and they become increasingly susceptible to further DNA damage. So it's kind of a spiral situation. Part of P53, P21's processing is the decision to route the cells toward apoptosis or no apoptosis. Apoptosis, programmed cell death. So there's activation of those enzymes involved in self-digestion of the cell if in fact there is damage that cannot be repaired. So in cells that have managed to get by the first round, you may have an ability to avoid apoptosis. You may have the ability to avoid immune surveillance. And at some point, you may end up with something called an activated telomerase. What happens every time a cell replicates? Which DNA? Where are the telomeres? Okay, at the ends of the chromosomes. Every time they get replicated, under normal conditions, they get shorter. Okay. Is telomerase active? Does telomerase do the cutting? No. In fact, telomerase lengthens telomeres. Make sure you got that distinction because it can be confusing. So if you've activated the telomerase, you're actually lengthening the telomeres each time you get a cell replication. Okay. So, let's see if I got a figure of what that involves. Okay, this is just a fancier picture of going from a proto-oncogene to an oncogene product and what can result in DNA damage, oncogenes, etc. I'm going to skip over this. Some of this was from my colleagues' postdoctoral work. BCL turns out to be one of those proteins that's involved in the apoptotic pathway, which is why she was concerned with it. It turns out that genes that are often involved in cancer formation are hotspots for primary or secondary mutations. I don't know if you've ever gotten any background on it, but during protein expression and during messenger RNA transcription, DNA, which is normally tightly wound around nucleosomes and around chromatin, tends to open up. Okay. There are some great electron micrographs that have been captured that actually show active areas of messenger RNA production and, in some cases, protein transcription. And what you see is kind of a bubble of activity around those areas of the gene or of the DNA that are actually active in transcription and translation. And so it turns out that those turn out to be hotspots for a lot of mutations. So it kind of stands to reason. The DNA is opened up. It's potentially vulnerable to insult by chemicals, by radiation. It doesn't have the protection of its normal chromosome. It turns out, interestingly, that among those hotspots are tumor suppressors, so P53, RB, P21. Do these look familiar? Okay. They're the elements involved in normal hormonal activity. Let's see if we've got a list of them. It turns out, if you look, there are a number of lists. This one happens to come from Sigma Aldrich, a chemical company that's compiled some of this data. If you look through the list, you're going to find tumor suppressor genes. Here's the ones we talked about already. Here's Wilms tumor, or WT1, very important in development. NF1, also developmentally important. APC, which turns out to be very important in the formation of colorectal cancer. These you may have heard of before, BRCA1 and 2, involved in breast and reproductive cancers. P10. Let's go to another list. Here, tyrosine kinases. What are tyrosine kinases involved in? They're transducers for what kind of pathways? Cytokines, growth factors. Okay. Serine-threonine kinases. G proteins. These are largely small G proteins. Gene rearrangement proteins. And nuclear proteins, these are transcription factors. June, Foss, if you're a neurobiologist, you're very interested in activation of these following activation of cells, neural activation of cells. MIB, MIC. Okay. DNA damage responses to any of these insults. Cell cycle activation at P53, P21, transcriptional program activation, so you turn on a bunch of other genes, you get DNA repair, and if it's bad enough, you can also route the cell toward apoptosis. So the process of carcinogenesis, inappropriately controlled cell replication leading to disruption of normal physiology, metabolism or structure, ultimately disrupting homeostasis irreversibly. A lot of what happens in the late stages of cancer are physical disruptions of the blood vessels, the nervous system, the endocrine system, the gut, the vascular system. They're actually physical disruptions. You also get disruptions of homeostasis. Cancer cells tend to have a high metabolic rate. They put off a lot of CO2 and decrease blood pH. Okay? So you get acidification of the system. And if it goes far enough, you will kill the system. Okay? Neoplasm, new cell growth, benign or not, you've seen that before. Malignant cancers leading toward metastases. We went through those hypertrophy, elevated cell size, or elevated hyperplasia, elevated cell number. Okay. This is the classical progression of cells toward metastases, final stages of cancer. So you have an induction or initiation event, some primary mutation, and it has to get fixed into the genome. So the cell that has that first insult, if it gets routed to apoptosis effectively, it will never show up beyond this point, this first point. If in fact it gets fixed into the genome, you may have avoidance of apoptosis and immune rejection, and you may become susceptible to some sort of promotion where you stimulate cell expansion of the mutated clone. Things like expression, self-expression of growth factors, or a mutation in which, for instance, a growth factor receptor no longer requires the binding of the growth factor to stimulate cell division. That does happen, and under those circumstances, you're going to expand that mutated clone. You're continuing these two elements during promotion. This is where some hormones may also be involved. Estrogen is a mitogen. It stimulates cell division. And it may come into, if it's still effective in stimulating cell division on a mutated cell, it's going to be a promoter. Ultimately, you get conversion or transformation, where you get secondary mutation, activation of telomerase, loss of cell contact inhibition, and you get something like expression of angiogenic factors. Angiogenic, what is an angiogenesis? Yeah. Not just veins. Blood vessels. Stimulation of the growth of blood vessels. And it's not so much veins as it is arteries. Why do I say that's important? What do arteries provide tissues? Oxygen and? Other than blood. What else? What's in blood that they provide? Not just hormones. Pardon? Not just white blood cells. More important. No, plasma, it's contained in plasma. Nutrients, okay? Cells get their nutrients, including oxygen, from arterial blood supply. What do you get in venous blood supply? Used up stuff, okay? So metabolites, waste products, that's the stuff that's going toward the liver, toward the kidneys, toward the outside, okay? You're trying to get rid of waste. Okay? But these cells may start producing their own angiogenic factors. Tumors, even particularly primary tumors, that grow, if they're undergoing uncontrolled cell growth, they get bigger and bigger, and unless they're making angiogenic factors, what happens? Can you imagine what happens? So you've got this mass of cells, it's growing fast, it's continuing to grow and grow and grow. What's it missing? It's missing oxygen, it's missing blood supply, and it's missing nutrients. What do you think happens to the cells that are farthest away from the blood supply? They're going to die, okay? They're going to die off. So you often get a necrotic center in tumors, primary tumors, secondary tumors. So those tumors that don't make angiogenic factors and don't get a vital blood supply, you're going to see essentially a rim of live, actively growing tissue around a dead center. As these things undergo this transformation, they continue to progress. You get tertiary mutations, and at some point, you're going to get small pieces, individual cells, breaking off, getting into the blood supply, and being carried throughout the body. Now, I think we talked about the fact that, say, in the pituitary or in small capillaries, cells go through one by one, right? Red blood cells, are red blood cells big cells or small cells? They're pretty small cells. They're about 15 microns across. Okay? Are they bigger or smaller than the average somatic cell? Smaller. An average somatic cell, 20 to 50 microns across. So what do you think happens if pieces of a tumor or individual cells from a tumor get into the blood supply and get into a capillary bed?
They're going to block it.
They're going to block it. They're going to get stuck there. And in matter of fact, that happens a lot. Now, in addition to some of this other stuff that they're producing beforehand, you'll often see that cells getting to this stage or into this stage are also expressing the particular proteins on their surfaces that are necessary to bind to or home to particular organs. They may also be expressing or producing proteolytic enzymes that allow them to breach the vascular the endothelium, the vascular supply wall, and therefore actually invade into secondary sites. Okay. So you end up with secondary tumors and ultimately tumors all over the place. So there's changes that occur at the gene level, at the subcellular level, cell level, tissue, organ, apparatus in the organism. So lots of different levels of change that are occurring in cancer. This is getting into the clonal selection idea, where you go from a cell where you start. It's normally undergoing self-renewal. Occasionally you'll get death or terminal differentiation. If you get an initiated cell or a mutation, you can begin essentially a second clonal line of cells. And you can imagine what happens if, in fact, you get selective clonal expansion of initiated cells. Let's say some of these expand first, or you get secondary or progressive mutations which favor growth of that clone versus growth of another clone, or you get some sort of malignant conversion of the initiated cell so that this cell and its progeny now will in fact seed secondary sites. I think I've got another picture here. This is another description of the same thing. This is the one I wanted to get to. This is a ball diagram of Noel's hypothesis. Noel's hypothesis is talking about the cascade of genetic damage and the cellular changes that ultimately lead from that initiation event all the way to the status of metastases and full-blown cancer. Let's see if I've got, again, initiation, promotion, progression. Okay. And selection. Okay. Notice here we've got the initiating agents are familiar to you. The promoting agents, inflammation hormones, estrogen, chemicals, growth factors, and growth inhibitors play a role here as well. So the balance determines the rate of promotion of these cells in their selection for rapid growth. Okay, another picture of the same thing. More of the same. I guess we got carried away.
Professor?
Yes.
I got the previous diagram, the ball diagram.
Yeah.
After the ball branches out from cell damage, and there are a series of balls that terminate before leading to another, before one of the balls go into more cell damage.
Yeah.
Now those are damaged cells. What are they corrected? Is it assumed that they're corrected so that they don't develop the cancer, or what?
What the diagram leaves out, it's a simple. Come on, previous. Oh, there we go. What it leaves out is the fact that at any of these stages, you may have elimination of some cells or some subclones. So it may end there. Okay? If, in fact, it goes on, it's going to form the next stage. You notice when this goes to blue, I think, you got some lineages that stop, okay, where you may have a static point where apoptosis takes over or immune suppression takes over, so those don't go any further. But there's a subclone that may be able to get to the next step. And part of what I'm kind of rushing through to try and get to is a description, here it is, a diagram which essentially talks about all the various steps that cells have to progress through in order to go from that initial normal and then mutated cells to the point of actually being metastatic. There's multiple steps. And part of the reason cancer is a rare condition is because those cells have to progress through all of those steps to get from starting point to the end condition. And you can bet that there's attrition at each level. Okay? So, you've got, let's see, where we got? I'm trying to look where we got. Oh, that's the end point. Okay, you've got normal cells getting to the point where you've got evading apoptosis, self-sufficiency and growth factors. So they make their own growth factors and respond to them. An autocrine type stimulus. A lot of breast cancer cells make estrogen and they respond to it by dividing. Okay? Insensitivity to anti-growth signals. So they may lose certain receptors that would normally suppress their growth. You have to get sustained angiogenesis if you've got a tumor. Otherwise, the tumor dies off except for that dividing rim. Telomerase has to be activated. Now, has anybody grown cells in culture other than yeast? E. coli, that's bacterial cell. Mammalian cells in culture, I should say.
I have a mammalian cell.
Okay, you did mammalian cells in culture. What do you have to do about every 48 to 72 hours?
You have to passage them, otherwise they will start dying.
Okay, what do you mean passage them?
You have to subculture them, like you have to basically do the same thing, you change the media, you divide them.
Okay, so you take the cells that are there, you lift them off the culture dish, you dilute them and put them back at a lower density into plates with new media. It's called passaging. Has anyone ever heard the term the Hayflick limit? Possibly not. Yeah.
It's kind of hazy, but I think it's like the number of times a cell can be produced before it no longer has telomeres to support reproduction.
Well, it's not just telomeres, but telomeres is a big part of it. If you, in fact, go through doublings, so passage number, okay, over time, or I shouldn't say over time, it's passage number. In some cells, they only passage a few times and they'll die off. Ultimately, though, you may be able to get through a certain number, but eventually this is going to flatten out. That's the Hayflick limit. So this is actually number passages. And this would be I guess frequency. The point is you get to a maximum number of passages for the cells, and this is in large part because of telomerase. So lack of active telomerase. That's that. Now, there's one cell type I can think of that's been in culture since 1951 and continues to be passaged in labs throughout the world. Yes. HeLa cells. Okay. Where did HeLa cells come from? Henrietta Lacks, who was a black woman in the Baltimore area who had cervical cancer that was biopsied in 1951. And there's a whole story about it. If you haven't read the book on Henrietta Lacks, it's a good read. It's a good tale. And anybody going into medicine, there's another book for you. Okay. The thing about HeLa cells, though, is that they don't have, there's no Hayflick limit. So those are cells that have limitless potential to replicate. They're part of the reason we know about that particular barrier that normally affects cells on their way from mutagenesis toward cancer and toward metastases. But you've got all these steps that cells have to go through before you get full-blown metastatic cancer. Okay. This is just pretty much the same tale several times over. There are a number of, or a couple of instances that we've included at the end of this particular PowerPoint series, one of which talks about the development of the colon cancer. Another one talks about development of breast cancer. Let's see which one of them we got here. Here, this one's lung cancer. And if you're looking at genes that are overexpressed in lung cancers, there's this whole list. You notice that whole list includes most of the ones that we saw on that earlier table. So this would be research underlying part of the reason you saw them on the table. Initiation. We talked about initiation. You also can get initiation from something called inversion. One of the most common ones is a Philadelphia chromosome formation, and it's interesting because a piece in this situation, there is an enzyme down here, ABL, and there is a gene that's normally associated with apoptosis on another chromosome, and if you swap this piece onto that other chromosome and swap the other tail down here, you get what's called a fusion product between BCR and ABL2, or in ABL. The cellular ABL oncogene from the distal tip, when translocated to BCR, is found in 95% of cases of chronic myelogenic leukemia. So lots of leukemias arriving from white cells are in fact associated with this swap. The product is a chimeric mRNA and fusion protein that localizes the cytoplasm and remains constitutively active in phosphorylating STAT5 and suppressing apoptosis. So there's a case where you get suppression of apoptosis. This is a good picture of what's happening in moving from the primary tumor toward a metastatic condition, where you've got VEGF production. That's a local. That's a growth factor, vascular endothelial growth factor, being produced by the tumor, which it's responding to. It's also stimulating the ingrowth of blood cells into the tumor itself. And once you've got that condition, you can get cells breaking off, getting into that blood vasculature, and moving through circulation to secondary sites. Okay. It turns out that VEGF has a large number of family members that relate. Remember, early in the term we talked about the promiscuity of some hormones for other receptors. Well, here's a good picture of it, and here's the number of transduction factors that can be activated in those pathways. No surprise why it's stimulating growth. Okay, this is again showing you the pathway and getting cells to where you start with the initial tumor. It often becomes hypoxic. You increase blood vessel formation and ingrowth, proliferation. You get certain metalloproteases allowing the breach of the cell membrane, the vessel membrane, and you get endothelial cell migration and adhesion, vascular growth and tumor growth, and then tumor cells getting out into the blood supply and metastasizing to other sites where you repeat the process. Okay. Progression, I'll pass that one. Okay. Chemical resistance to drugs. One of the big problems in treating cancers is that, well, first of all, has anybody read enough to know what kinds of drugs are often used to treat cancers? Anybody? Pardon? Cisplatin. Any other ones? There are going to be a number of you folks who have relatives who have cancer. What are they being treated with? Chemotherapy. What's chemotherapy?
Kills fast growing.
Pardon?
Kills fast growing cells.
Okay. Cisplatin and other drugs, often nitrogen mustard derivatives, like nerve gas derivatives, suppress the growth of rapidly growing cells. Did I say cancer cells? Okay. So what do those drugs also suppress the growth of? All other cells that are rapidly growing. Give me some examples. Hair. Nails. The lining of the gut. How about blood cell formation? Males, you should speak up here, because one of the most rapidly developing cells in the body are sperm. Okay? So sperm production compromised. Anything else? How about this stuff? Okay. The basal layers of the skin compromised. All of those tissues that are rapidly growing are going to be sensitive to a good many of the drugs that are used to suppress cancer growth. Okay. That's called non-specificity. That's a big, big, big problem. How about radiation? Radiation treatment is another commonly used treatment. Is that any better? Why? Okay. It also causes damage to cells. The one reason it may be a little bit better is because it tends to be focused on a primary tumor, a primary location, rather than being put into circulation where it can affect absolutely everything. Okay. Okay. Now, there is a tremendous amount of work that's been going on for the last 15 years and is going on continuing to try and make drugs for treatment of cancer more specific. Pardon?
It's like personalized medicine.
That's what personalized medicine in the context of cancer is all about. It's making treatment that is specific to a particular tumor cell type. Is there a metabolic pathway that's only going to show up in that tumor? Is there a receptor that's overexpressed in the tumor, making it more susceptible?
But isn't one of the reasons why cancer is just so deadly is because they become specialized, they can metastasize in any tissue type?
Well, tumors tend to be what are called archaic cells. They tend to regress in their expression of proteins. That's one of the reasons why they make the subunits of chorionic gonadotropin, which we talked about. That turns out to be a very archaic protein. It's made by some of the earliest stages during cell development. So it's like you turn back the clock on the expression of particular proteins. So that's part of it. But a lot of the work going on now has to do with targeting of chemical treatments to particular cell types. Personalized medicine. A lot of personalized medicine is also focused on the activation of the individual's own immune system. Anyone here ever hear of Jimmy Carter? Okay? Who was he? He was president of the United States from
77.
Okay, from 1980 to 1984. Okay, presidential elections are only held in even-numbered years. Okay, but he was president for a period of time. He has since gone on and done a lot of humanitarian work. He's probably better known now for his humanitarian work and the fact that he got a Nobel Peace Prize than he was for what he did as president. But he did something about 3 years ago that was also remarkable. Did anybody hear about it?
Well,
He had had cancer several times before, but what kind of cancer did he beat? No. Fulminant brain cancer. Okay? He had brain cancer. Okay? And what they did was he essentially stepped forward and said, I'll be a guinea pig for trying activation of the immune system. And in fact, it worked. Okay? Now, this was a man in his 90s. Okay? He's 96 or 97 now.
He's almost 100.
Yeah. He's been around a while. And he's still around. He's still building houses.
Sure is.
At any rate, the point is that activation of the immune system, so self-defense against the metastatic cancer, is something that's beginning to be a reality. It's beginning to be a key element of personalized medicine. Okay. So a little bit more. Okay, this is showing there are a couple of markers that you often see in breast cancer. Her HER2 is a receptor, Herceptin receptor, and it can show up in the tissues and make them resistant to Herceptin, which is a drug. You also get estrogen and progesterone receptors in breast cancer developing to a greater degree than you would normal. And those have all become markers of what kind of, what stage of breast cancer you have. If you are, if you express those, you have the potential to use those drugs as targets for specific treatment. If they are negative, the tumors are negative, you don't have those handles. And so individuals who are negative in all those receptors are hard to manage and often die as a result. Okay. I'm going to skip over this. Let's see if I can get to. There are several slides in here showing you what diagnostic stages of cancer biology would look like. And they go from stage 0 to, I believe it's stage 4. And it gives you some of the outlooks and where the particular stages will occur within the breast cancer patient. There's staging and classification. Stage 1 is the primary tumor. Stage 2 involves adjacent nodes of the lymphatic system, and then you get movement to other nodes, and finally full-blown metastases. This is with appropriate screening via mammography and self-examination. Usually you can catch it in stage 1 or early stage 2. If it goes beyond that, it becomes much more difficult to treat. And once it gets into stage 4, you'd better have your paperwork done because you're going to be dying soon. That's not funny. Okay. One of the things I wanted to get to was this particular slide from the National Cancer Institute. This was a kind of a compilation of work that was done at Johns Hopkins University by Bert Vogelstein and company. And what they effectively showed was that polyps that form in the colon of particularly aging individuals have different expression of tumor markers. And you get, in the early stages, you get very few mutations occurring. Stage 1, a lot more mutations. Stage 3, even more, or 2, even more. Stage 3, even more, and stage 4, even more. So this work is largely responsible for our understanding of this subclonal expansion, Noel's hypothesis, on how tumor cells are actually being formed. So this was work, like I say, on colon cancer. And it was neat because you can go in with a colonoscope and actually pluck off these various stages if you can see them on the wall of the colon. And you can take these, put them in culture, and ask all sorts of questions about them. So it was kind of a neat set of experiments as well as being important. And they found that you could, in fact, slow things down considerably if you're able to trim these off, cut them off surgically, which you can do with a colonoscope and associated curette. There's other video links for some of this material associated with that set of slides. Okay, so you should now be able to answer this question. and this question. There's plenty of other linkages to endocrinology. And because you've got so many different kinds of cancers that are linked to steroid production, for instance, or responding to steroid production, or growth factor production. Next time, I want to begin the process of going through steroidogenesis. So we're going to talk about